ahead and just announce that what we'll do for the final assignment is, as we sort of mentioned last time, we'll have people come up with a like mini project or experiment that you want to use some of the techniques we've covered in class, maybe in combination, and talk about what are the advantages and disadvantages of that technique, and based on whatever your disadvantages are, what might be an alternative or a solution to that disadvantage. And I think for efficiency's sake, we'll probably have you guys pair up, and you're all adults, so we'll allow you to choose your pairs. We're not going to pair you up. And then the presentations will be probably about 15 minutes. We have a 2-hour session for the 10 of you, so it'll be 5 groups. We'll have 15-minute presentations, plus that'll leave time. Oh, I didn't realize Jocelyn wasn't here. That'll leave you guys time, 5 minutes or so for questions, and then it will also give us time to transition, because who knows how clunky it's going to be trying to get everyone to share screens and all that stuff. And so for that purpose also, we'll probably create a different link just for that day so that it's easier for us to allow you guys to share screens as necessary. So we'll probably make like an official assignment write-up and distribute that like either tomorrow or Monday. Does that sound okay for everybody?
Sounds good.
All right, so I had just a little bit of an addendum to last time's talk based on questions you guys had and just going into more detail on some of the things that we talked about, and then this will give people time to join for the next talk. So I'll just go through these things quickly based on stuff that came up in the course of the talk. So I didn't go in much to actually the structure of the episomal concatenamers that we had sort of started to talk about regarding how AAVs are stably expressed over the long term. And it's because, in fact, for the recombinant AAVs, integration into the genome is suppressed. And so what does that look like? So the single-stranded DNA has these ITR structures that we talked about, which is the obligate area genomic region to allow our AAV expression. And so what does that do inside a cell? So basically, because there's suppression of integration, they can either become monomers or they can become concatenamers. So several copies of one AAV can concatenamerize. And so this one here shows 3, but I think that it's not necessarily known how many can associate. And it's thought basically, this comes from a book, I think, Adeno-Associated Viruses. So I think the next slide. Yeah, so basically there was, we had talked last time about if you infect a cell with 2 different AAVs, you could get hybrid concatemerization. And so there was actually a study to test whether or not they could hybridize. So the first model would be that they monomerize with each other. And the second model could be that if you have 2 different locus RAAV constructs, that they could basically hybridize. And it currently isn't known exactly how concatemerization occurs, whether it's through the ITR, which is what this paper shows, or potentially through the free DNA ends, which another paper shows. But essentially, they can hybridize. So, and it appears to be time dependent. So in this case, they have 2 different constructs and they infect the same cells. So across time, they start to get expression of both. So you can see whether or not it shows GFP or the other construct using developed with alkaline phosphatase. So you can see after 14 days, they're only really getting expression of 1. But then as more time passes, they get more of these hybrid constructs. And so Actually, this has been taken advantage of. So I didn't actually know this before I started looking into the concatemer structure, but you can actually force them to, either through homologous recombination or through concatemerization, you can actually get AAVs, which we talked about had limited payload capacity. You can get them to associate either by having a homologous area, and then they can actually become longer sequences once they've infected the cells. So you can put two separate sequences into two separate AAVs and get them to either concatemerize or homologously recombined using different tricks. So, and there's some other tricks that they've used. So this one is just showing two, but I think they've shown in the retina, they can get up to 3 AAVs to associate. So much bigger strands of DNA, so bigger payloads, blah, blah, blah. And then there was a request for a discussion of C-trio. We'll talk a little bit about how the mechanisms work this time in terms of like the flex constructs and how pre-recombinase can be used in combination with FLIP. But just in terms of the tracing method, there's a tracing method called TRIO, tracing relationship of input and output. And then you can combine that with conditional expression, so you get cell type specific tracing relationship of input and output. And so these were constructs were reported in 2015. And in this case, they essentially have, they combine rabies, which allows presynaptic staining, as we talked about, or presynaptic expression of GFP, but they're also expressing a conditional construct that's given in the terminals. So in this case, they're using canine adenovirus that has retrograde transport. And so in this case, they're administering CRE into the terminals, and then they get, once that CRE is taken up into the cell body, they get expression of CRE-dependent constructs, the TVA and the G protein. Those start to express, which will allow the presynaptic uptake of GFP. And in this case, instead of CRE itself, they're using a CRE mouse and expressing FLIP. So then they get FLIP and CRE, and then basically they have an FRT, G and TVA, and then so the co-expression of, the FLIP construct is CRE dependent. So then in the CRE positive cells only, you get FLIP expression, which recombines TVA and G. We'll talk about these mechanisms in terms of intersectional genetics much more in depth. But basically, what does that look like in practice? So I didn't really love the schematics from the original paper from Leach and Lowe's group in 2015. Then a paper out of Catherine Dulac's lab in 2018 showed that they could use, here, this is the C-trio. You can choose different sites of projection targets of your cells of interest. In this case, it's a galanin CRE mouse, so galanin expressing cells from the medial preoptic area are the cells of interest. So we want to know, based on where those cells project, what are the inputs. And so you can choose to put your conditionally expressed FLIP into different projection targets, allow that to be retrogradely transported, and then you have your FRT dependent expression of TVA and RG, and now those will only be expressing FLIP in cells that are CRE positive, and then you go in basically with your conditional rabies constructs, the TVA and RG that are necessary for the delta G in the rabies expression, and then you get the inputs. And so basically how that looks in terms of data is very confusing. So essentially you are getting the outputs that have specific inputs based on, in this case, a cell type population, the MPOA galanin neurons. And so there's most of this graph here is gray. So generally, so these are all compared to all gallinin neurons, all the rabies inputs, so just the traditional CRE-dependent TVA, RG, so total presynaptic input, and then we compare the input specific, the projection specific inputs. So these are all in comparison to that original tracing. So in some cases you get de-enriched, depleted inputs based on a specific projection, which means that there are certain inputs that are biased toward projecting to a certain output. So in the case of these neurons, there were some that were significantly different from total rabies tracing, but across these different ones, all these different projections that were examined, you know, it's like maybe half of them have significantly different input. So it's a lot of work for not a ton of information, but potentially it could be useful if you have, if you really want to manipulate the inputs and have some sort of specific output that you're looking at in terms of the downstream signaling. Does anybody have questions about that? I think that's all I have from last time. So then go here. Am I still sharing? Okay. All right, so today I'm just going to quickly go through lesions in pharmacology, more or less just to tell you what you can do and what the limitations of using those techniques are, because I think we need to really spend, I think for the purposes of this course, it's much more important to go through in detail genetic manipulations. I think that's what most people are trying to use, especially in terms of the tools of using chanorodopsin and GCamp. But a lot of these techniques, as we talked about with tracing and anatomy, you may be using this in conjunction, lesions of pharmacology, in conjunction with your GCamp or chanorodopsin manipulations. So I didn't want to completely abandon them. So as usual, I'll start with a little scientific adage. Scientists tested a frog, they cut its legs off and said, jump. The frog did not jump. Scientists therefore concluded that when frogs lose their legs, they become deaf. So this is just to highlight that, you know, we need to be really careful when we're interpreting our results based on what manipulations we're doing. So I've been spending a lot of time in quarantine, so I imagined what a reviewer would say about this. Obviously, you always have 3 peer reviewers. The first one says that you're over-interpreting your results. You need to provide a supplemental discussion of 5 pages in terms of the alternative interpretations, and be sure to cite the work of that reviewer. Reviewer 2 says, can't you use CRISPR for this instead of cutting off the mouse's legs? And then the third reviewer says you don't have appropriate controls. Frogs are also a terrible model organism if you're modeling human disease, and we're just going to reject this. So obviously I've been spending a lot of time in front of this computer, but also these are all valid points that are completely relevant to whatever manipulations you're doing, particularly lesions, but also in terms of pharmacology and potentially up to genetic manipulations. So you need to keep in mind the potential pitfalls when you're doing these manipulations, and really think about it at a systems level, what you're doing to the system when you make these manipulations. So again, we're going to talk today about lesions in pharmacology really quickly. We're going to very quickly go through knockout and knock-in, because most of the applications that you guys are probably going to use, you're going to use conditional expression. But a lot of times these are related to some of the knockout and knock-in technologies. I'm going to talk a lot about inducible expression, but it gets a little bit complex. And then I also want to specifically focus on tagging active cells. So this is going to be primarily discussed in the next lecture. And I may or may not have time to get to the CRISPR stuff, so that may go into the following lecture as well. But I can at least maybe give you an introduction to how CRISPR-based expression systems work. So first we have lesions. You guys recognize these famous examples of lesions. So in this case, we have, does anybody want to venture a guess?
To Phileas, I don't remember his first name or last name, but it was a worker who got the stick enter his, I mean, like exactly as described, and he just had some personality trouble after that, but he was able to function quote unquote normally.
Yeah,
it was Phineas Gage.
Yeah, this is Phineas Gage. And then you guys recognize this one?
That's the guy who lost, I forgot what they called him, and he's the guy that lost his entire hippocampus.
HM.
Yeah, HM.
Yeah.
Last it was removed.
Yeah.
It was removed.
Yeah, so there's some, actually, I think they just recently unanonymized HM, and then also all of the data is now released because they sectioned his brain, and there's this controversy now because in addition to the removal of hippocampus, he seems to have had some cortical damage, and now they're all up in the arms about how to interpret all of the lifetime of data from HM. But anyway, just an aside, but both of these, you know, these are both actual lesions in humans, and then these are also can highlight the concerns with interpreting lesion data. Of course, when we do it in the lab, we're a little bit more precise. So there are several methods for lesioning. You can do a physical lesion, knife cuts, and knife cuts were particularly popular for cutting fibers of passage. Aspiration, which is typically used actually in conjunction if you're going to put a lens in, for example, Bren lenses or cranial windows. Sometimes there's a little bit of aspirate. Actually, maybe cranial windows don't require aspiration, but you may have to aspirate tissue to get a lens in place in deep tissue. Electrolytic lesions, chemical lesions, there are a bunch of them. And then there are now some techniques where you can do viral mediated ablations, and these can have a little bit more cell specificity. So all of these have their own caveats and drawbacks and advantages and disadvantages. So basically there are some practical concerns. Again, going back to their first slide, there's a little bit of an issue with the interpretation of function. If you're trying to damage a brain area and say, oh, X was, the mouse couldn't do this behavior afterward. The question is, well, does that behavior live in that brain area? Is it just part of the circuit? Or is it, in the case of the frog, you cut off the legs? So there's some issues with interpretation. In the case of physical and electrical lesions and potentially chemical, I didn't have time to look it up. They destroy both the cell bodies and fibers of passage. So you're not just damaging cells that are in that area, but also any incoming or outgoing information. And then chemicals, some of them are very nasty. So you have to get a lot of approvals, and you have to be really careful when you're working with them, because obviously if they're killing neurons, you have neurons too, most of the same channels that the mice have. In this case, we're mostly talking about mice. And then for viral lesions, supposedly there are some fast acting ones, but I've heard through the grapevine that in practice that all viral lesions take at least a month to take place. So then you're holding on to your mice for a long time. In particular this, as well as DTA and the caspase-3 based methods. So then in terms of pharmacology, you can actually do reversible inhibitions. You can use these agents, musmol, lidocaine, and these are sometimes called reversible lesions, but I don't think inhibiting cells are necessarily the same as lesioning cells, because you could have different effects when you remove neurons from a circuit compared to when you just silence them. And then in addition to these reversible agents, you could also put, for example, agonists or antagonists for receptors, peptide receptors, hormone receptors, this type of thing. And so usually this is done through cannula system. So usually you implant a guide cannula into the brain, and then when you're ready to infuse, you put an internal cannula in attached to tubing, and then you infuse the drug, wait some amount of time for the drug to become active and test in behavior. I couldn't find a reference for it either, but I've also heard that when you have a guide cannula, you can get a bunch of scar tissue formed at the end of the guide cannula. And so when you're trying to do a drug infusion, the day before you're doing your drug, which also you need to do, your vehicle as well as your drug infusion, that you should insert the internal cannula, and it should have a projection length so that it punctures that scar tissue, because just simply the act of puncturing that scar tissue can already have a behavioral effect. So don't do that on the same day that you're trying to do your manipulation. Do that the day before so you get rid of that. I mean, I'm sure it builds back up, but remove that tissue. So of course there are going to be practical concerns with this as well. It's hard to tell how far a drug diffuses from your injection site. And obviously there's going to be sort of a halo of drug concentration. So where is the drug active versus not active? And so some people have tried, so for example, this is mucimol conjugated to Botopi, but this is a fluorescent marker, which basically means that it's going to be lipophilic. It's not going to have the same diffusion properties that the chemical itself has. So you can try to estimate based on a fluorescent reporter, but you're not really going to know. And of course, you're not going to kill them at the peak of the drug concentration. You're probably going to kill them after you've done your behavior assessment. And this goes back also to the half-life of the drug. So you're going to have some sort of ramping up of the accumulation of the drug in the region you want and activity. Most drugs, I think, if you infuse them directly into the brain, it's on the order of, tens of minutes, maybe an hour at the most. And then you have a window of like an hour, maybe up to 4 hours, depending how concentrated the drug is and how effective it is. So the half-life of your drug can affect your behavioral readout, and then also the half-life of the drug. You may need to wait a lot of time before you try to do another test on that same animal, because if the half-life is particularly long, like 12 hours, you might not want to do a test the very next day. You might want to wait two or 3 days before you do a retest. But it is an advantage of pharmacology that you can do multiple testing on the same animal. So now we're going to move into the genetic manipulations, because I think we're going to spend a lot more time on that. And the dirty secret of this class is that we're mostly talking about mice. So I'm just going to, outright only talk about mice in this lecture for the most part. So why have people really developed mice? They have a fast generation time, 19 to 21 days gestation. There's a really good system for embryonic stem cell culturing that allows for making these mutant mice, lots of inbred strains. And so one of the things that is taken advantage of is that you can have an ES cell and a blastocyst from different strains of animals, so that you can create chimeric mice, and then that facilitates knowing if you've got your construct or not. Litter size is usually 5 to 10, it can be much bigger. So you get at least 5 animals every 3 weeks. Of course, female estrus cycle is 4 to 5 days. I only mention this because it seems like a lot of people aren't aware of how long female estrus is. And then you can actually check the estrus cycle, just kind of for your information. You can do, basically look at the cells that are in the vagina at different parts of this cycle, and basically stain them. And you can see diestrus, proestrus, estrus. So a female is receptive between proestrus and estrus, and mesestrus and diestrus, they're non-receptive. And then puberty is pretty early, so around 28 days in mice. So there are a bunch of different genome projects that are trying to sequence genes, the entire genome of different organisms. Of course, just to give you some background information, the first complete human genome assembly was released in 2003. Currently, they're on version 16C. The first draft mouse assembly was in 2001. I think I have later in the slides, I think they're on something like release 38 or something like this. You can find all of the information on these different websites. This is also a huge collaboration international across these different institutes. But I just wanted to point out that, you know, we're going to talk a little bit about how methods to manipulate mouse genes. And of course, having a genome assembly is not equivalent to having an annotated genome. So just because we know the sequences doesn't mean we know necessarily what's there. We know sort of open reading frame sequences and some promoters and like start sites and these kind of things. So you can sort of anticipate where genes should be, but they're not necessarily all annotated, which is why there's continuing efforts to, so basically the first human genome in draft mouse, this was like very low, like 1 to 10 humans or something. So now they're trying to sequence many, many people so that they can see all the SNPs and all the gene structure, see if they can get better alignment of sequences and that kind of thing. So of course, here's the mouse genome. As I said, they're on release 38.P6. Supposedly the 39th version is coming soon, early 2020, but just to kind of give you an idea, there's something around 22,000 coding genes. This number has actually gone up since the last release, non-coding genes. So I think this probably has to do with better annotation of what those genes are, pseudogenes, and then transcripts. So basically there's a place where you can go and identify genomic sequences if you're interested. But not all genes necessarily are completely annotated right now. And then of course, the point of using mice is that we're trying to, in most cases, not all cases, trying to model human disease or human condition, human biology. And so this is being done through the Mouse ENCODE project. And so basically they're comparing elements between mouse and human. And this was one of the figures from the paper they released in 2015. And here they're showing different tissue types in different colors. And they're showing mouse in the open squares and human in the filled in squares. And if they basically look at the sequences across two principal components, you can see that the human and mouse version of a lot of these genes expressed, tissue-specific gene expression really clusters between mouse and human. So I thought that was kind of cool. So they're also trying to compare where do transcription factors bind, how is the chromatin organized, when do cells divide, and what genes are important for different cell divisions during development. And a lot of it is conserved. So that's good news for us that work on mice. There are a lot of places where you can find knockout mice or pre-drivers. So the days are gone when a lot of people are making their own mice. So these are just a few of those places. Of course, the GenSat project trying to make pre-transgenics using the BACT technology, knockout mice, Gene Trap Consortium, and Gene Trap can knock out or find enhancers promoters. Obviously, a lot of mice are archived in Jackson, the MMRRC. I've never used IMPC, but it was another one I came across in preparation for this. So again, I'm just going to kind of go over quickly knockouts and knock-ins. I'm assuming you guys have encountered this information at some other point. But basically, for knockouts, you can either use homologous recombination to go into the endogenous gene site and insert some sort of disruptive sequence, in this case, neomycin. For gene trapping, you can actually put in a gene reporter, essentially, that has no promoter. And then if you happen to randomly integrate it underneath the promoter that you want, you get expression. So in one case, you need to know the sequence. In another case, you don't. Most of the time, in 2020, you're probably going to know what the sequence of a gene is. But when people started, mucking with genes, you didn't necessarily have a sequence to start with. So there's some issues with knockouts. 15% of them are embryonic lethal. So we can basically use conditional strategies now, which we'll talk about later, to get around this problem. Of course, nothing might happen. You might get no phenotype. That may be because there's genetic redundancy or compensation that masks any effects of your knockout. And then if the gene doesn't have exactly the same function or the same expression profile in a mouse that it does in a human, you may not recapitulate a human disorder. So there may be a human deletion of some genomic sequence and you knock out exactly the same sequence in mice and you may not get any of the same phenotypes. So then in terms of knock-ins, you're basically here again, you're going to use homologous recombination techniques to target an endogenous gene locus to insert material. So in this case, they're talking about LOXP sites, which will, oh, actually this is using LOXP to specifically as part of their construct. And basically you take your ES cells and then you use homologous recombination to put in your material beneath the native promoter that you have. So it's very similar to knockouts. And here again, you may not recapitulate a human disorder. A lot of times the problems with knock-ins is you may get mosaicism with expression too, because the native promoter may or may not be highly active in every cell type, cell tissue. So then in terms of transgenes, I actually couldn't find a cute cartoon for transgenes, but essentially they're primarily synthesized using bacterial artificial chromosomes. So these are just enormous plasmids that can contain 150 to 350 kb. So I thought I'd just tell you, I recently made a transgenic mouse, even though I told you that people don't really make mice anymore. So basically our objective was to, we had a urocortin-3 Cree mouse, but we wanted to make a urocortin mouse where we could express FLIPO. And so basically the idea was that we were going to take the back that contained the urocortin promoter regions, insert FLIPO with a cleavage site and a BFP tag into the urocortin-3 gene. So because there was already a GeneSat created Cree mouse, we were able to just basically piggyback off that because they had already, you know, cataloged what back clone they had used. They had all of the annotation in terms of where to, what the enzymatic sites were that you needed to get upstream of the start ATG for the first coding exon of urocortin-3. So in our case, urocortin-3 promoter is not defined. It's just all of the 5' UTR of this gene. So that's one advantage of using a transgene as opposed to like a virus. We wouldn't be able to put the promoter in a virus. So basically we just piggybacked off that. They used, I forget how to say RC something. Anyway, they basically used the 2 different LOXP sites, and we'll talk about how you can do that to exchange parts of the gene, and then inserted this FLIPO P2A tag BFP. So this was pretty easy because once we had this construct, we had all of the annotation information we needed. And then we sent this modified back to the genome modification core at Einstein, and they did the ES cell injection for us, and then we got 46 transgenic mice. One of them was FLIP positive, so that was interesting. But anyway, just to tell you, it's not so hard to make a transgene, but there are some issues with it. So with transgenes, there's the only homologous recombination that happens is when you're actually modifying the back. Once you are inserting the transgene, that goes in anywhere. So you can't necessarily control where the transgene integrates. There are preferences for it, where they'll integrate, usually in open chromatin sites, but you can't control it, and you can't control how many times it integrates. So that can also be a drawback to making transgenes, because you don't know how many copies are in there, and obviously the more copies you have, the more disrupted DNA you have. So this process of modifying embryonic stem cells won the Nobel Prize in 2007. These three researchers, Kopecky, Evans, and Smithies, they received it. And obviously all of these techniques that we've talked about so far rely on embryonic stem cells. So then after you've done all of the stuff you need to do to get the transfection, your targeting vector into a cell, then you screen them with positive and negative selection cassettes. So on some of the slides you probably saw neomycin. There are some other resistance cassettes you can put in to either positively or negatively select for genes that have appropriately incorporated your construct. And then once you have selected cells, you can inject those into a blastocyst. So usually there's a preferred strain that the ES cells come from, and the ES cells are cultured, and then of course the blastocysts are removed from supraovulated mice. And so then you can implant them into the mother, and then the first generation F1, well, actually I don't know if this is F1 or F0 or something, but you get a chimeric mouse, and then you cross it to a wild type mouse, and hopefully you get progeny that have your gene of interest. But it doesn't happen for all the chimeras. In fact, it only happens if there's germline transmission of your cells. So the cells in this blastocyst, some of them are going to be wild type and some of them are going to contain your targeting construct. So hopefully there's basically good parameters for what stage of a blastocyst to inject the ES cells in. So hopefully there's a good chance of germline transmission, but if you don't have germline transmission, you will not ultimately get a transgenic animal, or sorry, any kind of mutant mouse. So then all of your mice are going to be hybrids. So usually the ES cell is one strain of animal, and then the blastocyst comes from another strain of mouse. So you're going to want to back cross the animals because the strain is going to be hybrid. And then for back transgenics, another reason why you might want to back cross is to dilute any effect of having multiple transgenic insertions. And there's also a concept, in, especially when people are trying to do studies of like F2 generations, it's known that the F1 generation, so in the F1 generation, all the copies on one and all the copies on the other come from either mom or dad. So you don't have mixed expression. So it could be that within a locus, the copy from one strain or another strain, either mom or dad, is going to be super dominant over the other one. So as you back cross the mice, you mix those locuses so that you don't have competition for one or the other. And this may not be so important for other types of research, but if you're doing behavioral research and you have a strain that has like a super depressive phenotype or is really dumb and you're trying to use it to do learning tasks, you may want to back cross to a strain that is either easy to train or does not have a depressive phenotype, depending on what behaviors you want to study. So this part we're going to go into a lot more detail in terms of conditional expression systems. So this might be a good time to take a break before we get into a lot of the conditional expressions, or is it too early? Do you guys want to wait?
Oh, it's fine, especially if it's dense content after.
Yeah, so I think this is a good time to take a break before we get into the conditional systems, which are going to be a pretty big chunk of today's lecture. So we can take a 5-minute break till 11:16. Really go into much more detail on conditional expression. I just wanted to give you guys kind of an overview of the other techniques, and then we'll go into much more detail regarding conditional expression systems. So what does it mean that expression is conditional? So you're basically using genetic tricks to restrict a gene from being constitutively express. So whatever if it's a knockout, knock-in, transgene. So combining, a lot of times this will depend on having like transgenic expression of Cre recombinase or something like that, in addition to say like a knock-in. And in this case, like you can generate a knockout by knocking in LOXP sites to a endogenous gene locus. You can also combine conditional expression with viruses, which we talked a lot about viruses in the last lecture, but we'll talk about how you can use these tricks to get viruses to facilitate conditional expression. And then, as I mentioned, you can sort of use conditionals in conjunction with inducible expression to sort of both temporally and spatially restrict your gene expression. And so typically we're talking about Cre, but I'm going to go into a little bit of detail on some of the different systems that you can use for conditional expression. So first we're going to go through in quite a bit of detail the Cre LOX system. So this is Cre attached to its DNA recognition sites. So this is originally derived from the P1 bacteriophage. Here's the bacteriophage. And it was discovered in 1981 by Sternberg and Hamilton. So Cre stands for causes recombination. I also saw in my research cyclization recombination, but I don't think anybody really believes that Cre stands for that. I had a weird experience where I wrote a paper and we were using Cre, and they kept trying to ask me what does Cre stand for, and at one point they had put in like cyclic AMP recombination something. I was like, no, this is not cyclic AMP, it's something else. But now I know it stands for causes recombination. And then the LOXP site, so this is the actual enzyme. And then the sites that it recognizes are LOXP. And so LOXP stands for locus of crossing over, so Xing over, and it comes from bacteriophage P1. So some other terminology you might come across is floxed, and that means flanked by LOXP sites. And Cre recombinase, actually all of the recombinases that I'm going to go into detail on are tyrosine site-specific DNA recombination enzymes, and we'll talk about what the specific mechanisms of those are and how they work, because it feeds into how they can be used to do different things to the intervening DNA. And so another thing that's similar across the different systems that we'll talk about is the structure of the sites that are recognized. It's a 13 base pair palindromic region. So that means it's one direction one way, it's like a left hand and a right hand. And it flanks an 8 base pair spacer sequence. And this is conserved between Cre, FLIP, and DRE mediated systems. So we'll talk about all of those through the course of this section. So a lot of different enzymes that are important for DNA repair and end joining and this kind of thing are based on tyrosine recombination reactions. And so how that works is that there's 2 sites, and they basically have the enzyme will bind at both sites. And then in the case of most tyrosine recombinases that I've looked up, there's basically this sort of opposite mechanism of where the tyrosine will take enzymatic action to create these OH groups on opposite sides. And then because of this opposite orientation where the tyrosine will create this nick in the DNA backbone, creating this free hydroxy group, then they sort of cross over. So there's a long side here and a short side here, a short side here, a long side here. And they sort of cross over then to create this structure that's known as a holiday junction. And so so in this case, it's sort of annoying because it's 2 purple strands and 2 red strands, but here you can actually see in 4 different colors the different strands, the complementary strands for each side, how that holiday junction looks. And so basically you get this structure, and then the opposite tyrosines from the ones that were not these, now the opposite sides, become active. They make the same kind of nicks. You get the same sort of annealing. And now you have basically swapped the sides of the DNA. And so this is how most of them work. So this is just, Cre is just one kind of these. And so basically to go into a little more depth on Cre recombinase reaction, and so the motivation behind showing you guys these mechanisms is that a lot of times they're shown as just these little arrows in diagrams, and it's not that easy to understand why, oh, two become one. So I'm going to kind of go through a little bit in detail why that happens. So here you have the LOXP site that's 34 base pairs long. Here you have the palindromic sequences, 5 to 3, and then obviously they're complementary on the other side, and then you have the spacer sequence. And so here again, this is basically the similar diagram to what I just showed you, but here in the case, so sorry, I meant to say over here, that in the case of Cre recombinase, you're having binding of the enzyme on this side of the LOXP site and this. So you have two Cre binding sites on each LOXP site. And so that's how you get this association, strand exchange, the cleavage events that we talked about. All of it's the same as in the previous one. And here again, two enzymatic, basically pairs. And so what does that happen? How does that work? So I'm going to talk a little bit more about what the function of the spacer region is, but the spacer actually determines the orientation. So a lot of times you'll see it as an arrow. You're always going to be having two binding sites on each one. And the spacer sequence is going to determine which direction it goes. So that's why you see this little arrow in the very simplified diagrams. So the way that this pairing works between the opposites, and remember the opposite sides are active, that happens in a particular orientation. So when you have two of them in the same orientation, you get this type of association in this kind of tetramer that causes the reaction. And so this leads to annealing. And here, the reason that I like this diagram over the previous one is that you can actually see, because these sites are palindromic, when they do this crossing over switching, you still get the same LOXP site, but one of them came from the other side. So you get sort of this hybrid LOX site in the end. And so one of them is a hybrid like this, and one of them is like this. So that's why it sort of ends up that you started with two, and in this case, for excision, you end up with one. So one of them became a loop of excised DNA, and one of them now has a piece of each of the LOX sites here. And so if you have them in opposite orientations, so one is pointing to the left, one is pointing to the right, it's going to cause the sort of loop on this side such that the orientation is still the same as it was here. And so in this case, you're actually going to have an inversion of the gene of interest. And so in this case, again, you end up with the hybrid sites. In both cases, you end up with this like hybrid LOXP site. But instead of being AB, now you have BA, and it's in the antisense direction. So again, using pairs of LOXP sites, you can get this excision event. As we just talked about, you can get this formation to do inversions, and you can actually have LOX sites on separate strands and get sort of a translocation of DNA. And sometimes this kind of trick is used in the ES cell systems to get different DNA sequences when you're trying to mutate your site in ES cells. So a very simple way to use LOXP sites in pairs is tissue-specific knockouts. So I did a deep dive on PubMed, and I found the first tissue-specific knockout, and basically they had a T-cell promoter driving the expression of Cre, and then they actually had put LOXP sites around this endogenous gene, pol B, and basically got specific deletion of this gene, pol B, in T cells. And so, you know, this is just one of those things that I find fascinating, looking up these old papers. So it says, although these were the first in print with a tissue-specific knockout, about 100 labs were working on them. So I can just imagine what it would have been like to be a molecular biologist in the early 90s. Like, it must have been insane competition at the time, because this was not the only thing they were doing. There were people cloning different genes, all kinds of stuff. So highly competitive. So again, just very simply, another thing that you can do with LOXP sites that are in the same orientation is basically have it surrounding the DNA sequence for a stop codon, and have your gene of interest. And then in this case, you know, it's a cute little cartoon. You have some sort of heart-specific promoter, MYH6, and you get GFP expression in your tissue. So that's just the very first scratching the surface. So just to go into a little more detail about the actual genetic codes that we're talking about here. So again, we have the inverted repeat. So this is exactly opposite of this site. So this is the directionality. And then you have a spacer sequence, and the orientation of it determines the directionality. So if you have the same one, they're in the same direction. And again, this will lead to deletion, and that is a permanent deletion. We'll talk a little bit about why it's important that it's permanent. If you have it inverted with just a single sites going the opposite way, you get this flip-flopping back and forth because the enzyme will keep binding, you'll keep getting this association, and it just keeps kind of going back and forth. So I'll talk about how they've done some tricks using the sequences to get this to not be reversible. So that comes down to the spacer region. So in 1998, they were really describing what is the spacer region, what do the different domains do, and they found that you know, you have the two palindromic regions, but then this region in between is asymmetric. It contains two subdomains. The first has 2 to 5 base pairs, and the next has 6 to 7 and nucleotides. Sorry, I was like, what did I mean by NT? Sorry, I was making these slides late last night. So essentially here in this paper, having identified these two domains, they basically went through and did mutations at each one to figure out which were important for the cleavage, the activity of the Cre recombinase. So they identified that there's sort of two different domains. They're important for separate parts of the reaction with LOXP recombination, parts of that sort of biochemical process that we talked about. And so in the process of doing this, they made a synthetic LOXP sites. This particular paper developed the 2272 LOXP site. And basically when Cre recombinase will bind, it's only going to associate two LOXP sites that have the same spacer region. So the palindromic regions are the same anyway. And then the spacer region, it's only compatible to ones that are exactly the same. So the wild type LOXP site is incompatible with the synthetic LOXP site. So that led to the development of flex switches. So what does flex mean? And of course they have this complicated capitalization system that's due to the actual, you know, what it stands for, the acronym here. And of course, when you're trying to do this in Word, it never wants to put FLE capital with a little x, so it's very annoying. But it stands for flip excision. So as I mentioned in the previous slide, Cre recombinase only recombines two sites if the spacer is the same. So what they did in this paper, Schneck in 2003, was that they put two genes in opposite orientation, and they inverted the LOXP sites, but then they put them in different orders. So basically, you could have one reaction with the wild type Cre sites or one reaction with the mutated Cre sites. So you can have two different reactions that will happen. In one case, you'll get an inversion with one set of LOXP sites. And so you get, once it inverts, because these are alternating in sequence, once it does the reversal, you'll end up with one LOXP site between the mutant's sites. If it goes to the other reaction, you get an accumulation of the LOXP sites on this side, and the other will get floxed out. And so basically, you're left with only one LOXP site either way. It doesn't actually matter which reaction happens first. You're going to end up with an excision of one or the other, either the mutated LOX site or the wild type LOX site. And so that in essence is going to flip it. So it doesn't matter which pathway it goes through, you're going to end up with LAC-Z in the correct orientation and GFP in the opposite. And so in this case, with this flex EGFP construct, in the absence of Cre, they get expression of EGFP and no expression of LAC-Z. And then when they have Cre expressed, it flips into the other direction, and now you have no expression of GFP, and you do have expression of LAC-Z. Does that make sense to everybody? So basically now there's kind of a library of LOXP sites, because obviously it is powerful to start combining them. So now there's wild type LOX, LOX 511, as we mentioned, 2272, and then there are some of these other ones. And as you can see, they have different spacer regions, always with the palindromic left and right regions. So what are some of the applications of flex switches? So these are a lot more versatile than just using a single LOXP site. So again, we've already talked about tissue-specific gene expression. In the simple way, you just have removal of a stop codon, but you can do things a little bit more fancy with the flex switches. You can have inducible gene expression, which we'll go into in more detail, potentially in the next lecture. You can control multiple genes. So as I just showed you, they were switching GFP and LAC-Z from one orientation to the other. So you can imagine how you could control multiple genes in other settings. Obviously, you can do a knock-in. I'll show you how that works, and that basically uses two different LOXP sites on two different sequences. And then you kind of get that reaction because you have the matching pairs on different pieces of DNA. And then, of course, you can combine it with expression of AAV. So we talked about that a lot in the last lecture, using AAVs to express in specific cells. So some of the nomenclature you might encounter, especially if you're just looking through like a catalog of viruses, for example, is double flox with inverted orientations, and that'll turn your gene on. So as we just mentioned, you have sort of alternating sites, and they're facing each other. So inverted orientation, DIO. Almost nobody uses the term DO, but you do have these where you get it off. It will turn your gene off. So here you basically have, instead of having your gene flip the wrong way, it's flipped the correct way, and then it flips the wrong way when you have recombination. And so these were borrowed from this website. So as I mentioned, there are several different systems for inducible expression, and basically when we're talking about inducibles combined with conditionals, usually you're talking about inducible expression of Cre, and then Cre will go and associate with LOX sites. So basically there are two very popular inducible systems. I don't think they're the only ones, but they're the ones that we mostly use in mice. Tetracycline inducible, so that uses a tetracycline responsive elements where this tetracycline transactivator, in this case it's a reverse one, so it doesn't bind until it is in the presence of TET, which is delivered using docs, usually orally or injected or something like that. And then once that binds, it allows the tetracycline transactivator to bind to the tree element and drive gene expression. And as I said, usually you use CRE, and then CRE does something with a FLOX gene. And then you have tamoxifen inducible systems. So in this case, you have, for example, you can have a CRE that has a binding site for a mutated estrogen receptor. That estrogen receptor, when bound to tamoxifen, can translocate into the nucleus to drive gene expression. And we're going to talk about some specific applications of inducible gene expressions in lecture 10, so I'm not going to go into detail on those systems at the moment. But I will just tell you a funny story. During my qualifying exam, they asked me, how do you induce tetracycline gene expression? And I was like, oh, you just feed it to them in the water. And they were like, you clearly don't understand how this works. So anyway, it's doxycycline that you feed to the mice. So for control of multiple genes, I told you in the previous lecture that there are some genetic tricks used for brainbow. So I'm just going to walk through this example where basically they're using combinatorial expression of different fluorophores to create a bunch of different colors. And so very simply, what does one of the constructs look like? This is Brainbow 1.1. This was reported in 2007. So without CRE, you're going to get expression of orange fluorescent protein. And then you have these different orientation of 3 different LOX sites around red fluorescent protein, yellow fluorescent protein, and cyan. And so basically, as I told you, depending on the order, you're going to get basically random reactions will take place first. But based on a clever placement of your LOX sites, you're going to get expression of any of these 3. And so if one reaction takes place first, you're going to get red expressed, but not the others. In another case, you're going to get yellow expressed and not the others. And then in another case, you'll get CFP. And so these are basically expressed randomly based on whatever CRE reaction occurs first, association between pairs of sites. And so you can see with CRE, some cells are expressing yellow, some are expressing CFP, and some express YFP. So that's the basically 1.1 version. And all they did essentially was have 3 of these in a row. And so basically in the first case, you have random expression of C, Y, or R, and then that pairs with 2 and 3. So if you get 3, if 3 times it chooses C, and that's whatever the CRE recombinase leads to expression of C. If you have all 3, you get blue. If one of those becomes yellow, it's light blue, and so on and so forth. And so basically that's how you get expression. In this case, it's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different fluorophores. So you can imagine you could go on, but I'm sure a microscope can't actually resolve the colors at some point. But this is basically how they got expression of 10 different fluorescent channels. So it's a combination of C, Y, and R in random fashion expressed. So I could not find a better schematic for this, but essentially to knock in a gene. You can have your gene with these inverted sites, and you can have them be non-compatible. And then you can put in a construct containing your gene of interest, also with the inverted orientation. And then these LOXP sites will associate, these LOXP sites will associate, and you'll basically have a swap of your gene of interest for the selectable marker. And so basically the advantage of this kind of system, you could use a virus to express this, you could use another mouse line to express your gene of interest, but essentially you can use the same line to express a selectable marker. So you could imagine a lot of times we use the ROSA 26 locus because it's kind of an open piece of DNA that gets highly transcribed. So you could imagine having your selectable marker downstream of ROSA 26. You can have that as a transgene or as a knock-in. You can use the same one to deliver bunches of different genes of interest. So it becomes a versatile system to use for that, as opposed to making a knock-in that through the homologous recombination method that we talked about first. So as we have mentioned before, you can combine CRE-LOX expression systems with AAV. So this was the first report of doing this for neuroscience applications in 2008 in Journal of Neuroscience. So basically they had, the flex switch with channelrhodopsin that was tagged to mCherry. And then in this case, they could have expression cassette where basically flex led to expression and turned it off. Sorry, it was on and it turned off. So this is what flex 4 means. And then flex rev means that it turned it off. So in this case, it would turn it into the correct orientation in the presence of CRE. So it was normally not expressed. And then in the presence of CRE, you got expression of channelrhodopsin and the tag for mCherry. And so basically this platform is now what most people are using to get expression of channelrhodopsin and other things. DREADS and GCAMP and reporters, all kinds of stuff. So there's some caveats with CRE-LOX. This can happen with any time that you have a transgene. Your insertion of CRE may itself lead to a phenotype. So as I had mentioned before, you don't know where it's inserting, so it could disrupt an important gene. So you really have to make sure that does not happen. You may get CRE expression in another place. So it may be expressed where you think it should be, but also maybe some other tissue. So you always have to check where CRE is getting expressed. And I think with knock-ins, you have the same problem, but you can have mosaicism. So basically how well CRE is getting expressed. So you could have incomplete expression. So the CRE is just not getting expressed very well in some cells. But in the other case, you may just not have good enzymatic recombination. So in that case, it's a little bit of a different issue. But these are things that can happen when you're trying to use CRE-LOXP. So one of the caveat, this isn't like the best example, but it's just an example from my own research. You do have permanent recombination. So you have to imagine if you're crossing an animal that expresses CRE to a reporter, so in this case, lox stop lox tomato, basically in adult animals at postnatal day 56, the gene that I'm interested in, again, urocortin 3, is expressed in the perifornical area, maybe a little bit in medial amygdala, but you can see it's not expressed in striatum. But then when I crossed urocortin 3 CRE to lox stop lox tomato, despite the fact that it's not really expressed in adults, or at least not at very high levels, I still saw tons of recombination here. So it could be that there was developmental expression of urocortin-3. I looked in the developing brain atlas, doesn't seem like there's much expression before day 14, and most of it's restricted to the hypothalamus. So it could be leaky expression or just very low expression that doesn't get picked up by in situ hybridization, but it's expressed enough to drive expression of Cre. So any place that Cre is expressed, whether it was because your promoter was highly active or not highly active, you're going to get recombination. So that's most of what I have to say about Cre. Are there any other questions or comments about Cre recombinase systems? So another system that is used a lot is the FLIP FRT system. It was derived from Saccharomyces cerevisiae yeast. This is a picture of yeast up here. This recombinase was described in 1980 by Birch and Hicks at Cold Spring Harbor Labs. And then the FRT sites were really defined in 1986 by the Sadowski group. And like Cre recombinase, I don't have to go back through the reaction again, because Cre recombinase and FLIP recombinase are both tyrosine recombinases. And in fact, the FRT site is slightly different from LOXP. It has an extra 13 base pairs or 14 base pairs, but it seems like there's a upstream 13 base pair direct repeat and another isolated base pair that are not important for the recombination of FLIP associated with FRT sites. So it really can be 34 base pairs, just like LOXP. I just wanted to point out that FLP is what it's called, FLP recombinase, and we call it FLIP. And I was reading the original paper in 1980, and they just decided to call it FLP. There wasn't a lot of reason other than it flip-flopped the bacterial within that plasmid, the DNA in that plasmid. And so they said it flip-flops, and we called it FLP. So that is FLIP recombinase. So FLIP, part of the reason that was the motivation of identifying extra recombinases is partly because they did want to have multiple recombinase systems so that they could combine them, but also the idea was that FLIP recombinase, because it came from yeast, it was a eukaryotic genome element, that it might be better in tissue, but in fact it was not as good. So FLIP recombinase was optimally active at 30 degrees. So most of the time when you're working in animals, you're talking about 37 degrees. So there were some improvements made on FLIP. So basically the 8th generation FLIP-E made some improvements. They added some genetic elements. And so compared to wild type FLIP, which was not expressing very well at, this is beta-gal expression that's dependent on FLIP recombination. You can see that 37 degrees or 39 degrees, you get almost none. And then FLIP-E was an improvement. And then FLIP-E was further improved upon, such that now you're looking at the clones that are showing this beta-cal recombination. They're comparing it to some other integrases from phase. And basically in this report, they showed that this FLIP-O version was almost as good as Cre. So you could use Cre and FLIP-O and hopefully get similar levels of recombination and be able to use them together. So FRT sites are flexible, little pun humor for you. So basically similar to LOX. So again, here's the LOXP library. This one's a little bit different from the first one that I showed you. But basically you can mutate the spacer region. Again, it has that spacer region that determines the directionality of it. So you can mutate, have different versions of FRT sites. And again, based on how that spacer is, determines the orientation and which ones will pair up. So you can use flip sites pretty much analogously to the way flip and FRT sites, the same way that you would use Cree and LOXP sites. And so there's actually a third one that's really still kind of under active development, but I know people are starting to use it, DRE-ROX. So this was another phage, and it stands for D6 phage, site-specific recombinase, DRE. And ROX is the region of X-over instead of the locus of X-over, so basically a crossover. And it's actually very similar in terms of the palindromic regions and the spacer, but the spacer is a little bit shorter. But more or less, the biology here is still the same. It's tyrosine recombinase. It was discovered in 2004, and it doesn't cross over in terms of DRE recombinase doesn't work on LOX sites, and Cree doesn't work on ROX sites. And more recently, I've only ever seen DRE and ROX combinations, but I think there has been a recent report that there are synthetic ROX sites. So you can have different ones such that you can start to use the kind of flex type of switch for DRE-ROX recombination.
Could I make a quick comment on that, Nerdita? So for DRE-ROX, all the early testing showed that there's no interaction with Cree-LOX. Then the Allen Institute made a bunch of lines, and a number of them actually showed that there is some some of the DRE or like ROX lines get recombined by Cree and vice versa. And I think it's probably one of those things where it's like 0.1%, but over the course of like 2 months or something, it accumulates. So it turns out, so Cree and flip truly are orthogonal, but Cree and DRE really aren't 100% orthogonal. It's just something to keep in mind.
And that probably makes sense because Cree and DRE both came from phage, and maybe the fact that flip FRT came from a different system. So I think there is active research into identifying recombinases from other types of organisms. Because if they have a similar mechanism of action and they have similar biology. I found tons of information about Cree-LOX and almost nothing about flip FRT. I really had to dig for it. So I guess basically once they figured out Cree-LOXP, they were like, oh, this is the same. So it's not as in-depth study, the flip FRT stuff. So basically, this is an example where they did combine all three, Cree, flip, and DRE. But basically, now you can start to see how you can get intersectional expression. So as we already saw with the rainbow example, just using Cree by itself with these complicated, I guess, syntax of like directionality and orientation of your LOX sites and different mutated LOX sites, you can get control of multiple genes, but then here you can have your different recombinases expressed in different cell types, for example, and start to get intersectional expression. So maybe it's better to stick with just 2, but in this case, they did use 3. So basically they had stop code. In this case, it was kind of more of a simple design where they just had similar orientations and they just flanked stop codons to get expression of either TD tomato or GFP in certain cells, depending on if there was the presence of DRE or flip or Cree recombinase. So this was reported in 2015. And so here you can see they made different constructs, but essentially this one is the one listed on the top, this RFLTG. And so basically in this line, you have DRE expressed under a ubiquitous promoter, so it's expressed everywhere. And then you have flip expressed. And so you can see with the expression of DRE, you get removal of that stop codon, and then with FLIPI expression, you get removal of the next one. And so now you can get expression of tomato, but there's a stop here, so you don't get any expression of EGFP. Sorry, EGFP over here. And then in this line where you have DRE, FLIP, and now you also have CRE, so now you're going to be removing the stop codon upstream of the GFP, and now tomato is removed. So when you have all three, you have expression of green. And so this can get pretty, obviously you can see how you can start to use a lot of different tricks. Maybe 2 is better than 1, but you can start to see, you can have, I meant to look up, but I could not find a good schematic of all the different choices, but you can see how, you know, if you have inversions where you have 1 on or 1 off, just using CRE, that you can start to get really fancy. You can have intersectional expression where if both genes are on the gene of interest, if both promoters are active, you can have your gene off, you can have your gene on, you could have your gene swapped for a mutated copy. You can do all kinds of things. So this is just to give you kind of an introduction to all the different things that you might be able to do. And I couldn't get a very good schematic for all the types of things that are being done. So one thing that was reported just using simply a combination of CRE and FLIP is sort of sophisticated fate mapping. So with CRE, as I mentioned in the previous section, your expression is permanent. So anytime CRE comes on, even if it's transient or early in development, even if it shuts off again, the promoter that you're using to drive CRE, very quick, sorry, I'm having a child near me. So sophisticated fate mapping. So as I mentioned, you get recombination.
I took a video of Daddy's story.
Okay. So you get recombination that will lead to expression, removal of a stop codon in this case, and you get expression under a specific cell type of end cherry. So then you can basically sacrifice the animal at different time points and see where the cell lineage, where those cells are migrating to. In this case, with the dual recombinase-based genetic fate mapping, now you have one stop codon upstream of M cherry that's floxed, and another one that has FRT sites around it. And in this case, you're going to get, actually, I think there's even an additional element that's going to suppress. So basically, in the presence of CRE, you start to see expression of M cherry. And then when you have expression of the second gene, you get FLIPO recombination that leads to excision of the M cherry. So now you only get GFP. So ones that in cells that only had gene A, you still have red, but ones that had both now are green. So that's just one application of intersectional genetics. And then I actually got through this a lot faster than I thought I would. So I didn't have a chance to fully flesh out my CRISPR part, but I can start on the CRISPR stuff and we can go into more detail on CRISPR, because I think there are already some improvements on CRISPR from what we had seen in the past. Sorry, does anybody have any questions on intersectional techniques for now?
Hi, Anita. I have a question. So for the CRE, you showed like an example of excision or inversion, noting that with the inversion, there's just kind of like constantly going back and forth. Are there instances where that could be helpful, or is that something that you mentioned that people have improved upon that to make it more likely that it's permanent. I'm just curious if the reversible application has been used for anything cool.
Yeah, I don't think I've ever seen it used, especially in mice, where you really want to know what your manipulation is doing. Potentially in like cell cultures, maybe it would, I could see where you might, just a second.
Mm-hmm.
I can say something about it, and he is busy. So I'm not sure. I mean, so Brainbow is one example of where it's useful because you get this flipping back and forth, and then when you put the doxycycline back in, then it stops and it's locked in one orientation. So one thing I saw at SFN a few years ago I was very surprised by is if you have an AAV where you have the two in-pointing lock sites and it keeps flipping, then apparently you get very little gene expression off of that. I wouldn't naively think you get 50%, right? But it turns out you get apparently very little when it's flipping constantly.
Interesting.
Yeah, so yeah, if you already thought that transgenes were sort of old news, then you're really going to wonder why anybody would use transgene now that we have CRISPR. So I'm just going to briefly start to introduce CRISPR because I think it deserves a little more attention than the time we have left. And I can talk about CRISPR and then also the inducible strategies next time. So basically, this is an element that was recently discovered and started really being used in mice around 2012. So it's CRISPR stands for clustered regularly interspaced short palindromic repeats. And the sites bind using the Cas9 protein. So this is CRISPR-associated protein 9. And based on a very short sequence, it can be site-specific. And because it is specific, and you can basically use a mouse strain that expresses, so obviously this is from bacterial systems, so if you have a mouse strain that expresses Cas9, you can start to multiplex just putting in different guide RNAs, which I'll talk about in the next slide, to really multiplex and manipulate multiple genes at the same time. And again, you're not randomly, hopefully you're not randomly, you have to check, but hopefully you're specifically modifying DNA so you don't have the problems of transgenic expression, and also because CRISPR is sort of a direct mechanism, you don't have to do complicated homologous recombination, and you can directly modify ES cells, so it's more, it has less of a step in the process of modifying your gene. You don't have to modify something and then put it in. You can use CRISPR to directly modify the ES cells, and this basically can speed up the traditional production of knockouts and knock-ins that we talked about in the first part. So here's the actual structure for CRISPR. So you have your Cas9 protein, and it has these sites where you're actually getting a double-stranded break, and then the Cas is positioned based on this short guide RNA sequence, and I can talk a little bit more the next time about the length of this and matching your genomic sequence and how specific that can be. And then more or less, once you have this double-stranded break, the double-stranded break is repaired using endogenous cell machinery. And so it can either go through the pathway of non-homologous end joining or through homologous recombination. And so basically my understanding of like the first generation of CRISPR is that this, this double-stranded break essentially was biased towards non-homologous end joining. And so that led to more likely that you were gonna get a knockout of gene instead of what you were supposedly supposed to be able to knock things in, but apparently that wasn't very efficient. So I can talk next time about some of the changes that they've made to the system that can bias towards this arm of the pathway as opposed to that one. And I didn't have time to get into the PAM sequences and all of that this time, so I can talk more in detail about that next time. So that was the end of that. I really thought that was going to take a lot more time to explain. So, but if you guys have any questions or things you want me to go into more detail on for the next class, so the next class I'm going to talk about using inducibles to tag active cell populations. And I can go into more detail about uses of CRISPR in mice. Anything else? I think we were also potentially going to talk about single cell sequencing if you guys are interested, but I know someone had also mentioned brain clearing. Although we have 2 more lectures and I'm not sure we can actually do an ImageJ demo for the second lecture next week.
Why not though? Because during the Python class, we all have Python on and then we do the manipulation at the same time as the instructor. So why couldn't we do the ImageJ?
I have to test it. I'll test it and see what I can do. I'm worried about the size of the images that I open. I'm worried that trying to broadcast the screen and open big files. So I'll either have to, I might be able to find a way to make the file sizes smaller, but usually the programs that I typically run with ImageJ use TIFFs because you need to be able to split the channels. So the files end up being really big. So I'm just worried it will crash my system. So I'll give it a test and see if it works. Otherwise, I can always provide you guys some simple code and some images that you can sort of play with offline.
I think the single cell sequencing would be good to go over, at least briefly.
All right. So that's all I have for this time, and we'll go through the other things in more detail, and then we'll see what happens in terms of the final class. And then we'll give you guys notice for those of you that are enrolled for credit. We'll give you guys a specific prompt for your project, and those will be groups of 2 to present the final project, and we'll do a separate Zoom link, so look out for that for the May 5th session. All right. Have a good weekend.
Thank you.
Thank you.